Thanks very much, Rick. I wonder if it might make most sense actually to have all of you, actually all of the afternoon speakers come over to the table and we can entertain questions both individually and as a group. And Rick, while people are assembling, I had a specific question for you, as long as you can find your name and listen at the same time. Actually, oh yeah. You still have the lavalier mic? Could you, yeah, that works for you. Thanks. So Rick, just maybe as a clarification for the experiment with the use of trimsulfa, it was given to the pregnant mice, latent pregnancy, I suppose. So could some of this be just interference with, now that I'm thinking about it, with folic acid synthesis?
We thought about that. We thought about that, but we and I'm trying to remember my reasoning at the time. It's late in the day for me. But we felt that, to summarize the internal discussion in the group, that we think that folic acid, you predict the folic acid would exactly have the opposite effect, and it would actually prevent the inflammation rather than promoting it. So we did think about that. There's no other explanation other than that deduction.
And again, to clarify, you said folic acid or folinic acid induces methylation of the gene?
So folinic acid induces methylation.
And then that increases expression or decreases?
The folinic acid increases expression.
Okay.
Possible,
right. Are there other questions? Margaret.
So one of the things that I've been really impressed with today, beginning with Marty's talk and finishing with your talk, is how very critical the first period of the mammalian life, post-embryonic life is. And I think I'm going to bring up, I might cause a riot or bring up something extremely controversial, but one of the things that strikes me is that in the first, there seems to be an increase in IBD in young children in the last several years, a couple decades. And one of the things that I think about is decades ago, or in a previous generation, the children were not vaccinated until a couple of years into life. And is there any, in mice, for instance, have you looked at any effects of vaccinating them on, like right as they pop out, on all of the things that you talked about? In other words, we're obviously doing something quite different right now that is trifling with the maturation of the natural maturation of the immune system. That's the obvious one that comes to my mind. So.
Skip is advocating to take this one, so okay. I can start. Okay. I can go first. Thank you. No, we didn't vaccinate our mice, and I'm reluctant to, that's obviously been very controversial whether vaccination has any adverse effect and causing susceptibility to a variety of diseases, and it's been hypothesized that vaccination to measles virus, as you know, may lead to susceptibility to inflammatory bowel disease and other things. But I think that most people believe that's probably not the case. But whether, it's interesting that whether vaccination, again, it's just pure speculation whether by vaccination we're affecting any core.
Well, I don't think vaccination early.
Yeah. Yeah, I think that there's no evidence. It would be pure speculation, and maybe Skip knows some evidence, but the vaccine, and I think you have to think very specifically about the vaccines that we're actually giving for the particular organisms, and polio and so forth. And it's hard for me to believe that there would be any effect on the early exposures to the particular those events, but I really can't speak to it any more than that on my own.
So so I think it's a great question, and there is no evidence one way or the other. But what I would point out is that you've only asked half of the question, because at the same time that we began to vaccinate people earlier and earlier, we cleaned up the water supply. And so, for example, polio, there were no epidemics before the early 1900s. Part of the reason was that children who get polio very early reset their bodies, and they do not get paralyzed. So the paralytic polio rate for children who acquire virulent polio under the age of 1 year is 0.05% or 0.02%. But if you let them become non-immune until they're teenagers, the paralytic polio rate, which is why we had epidemics in the 40s and 50s here that were so dramatic, goes up to somewhere between 15 and 20%. So what I would point out is that we're shifting not just the vaccination of young people, but we're shifting our entire total exposure to bacteria, viruses, very likely other things which are in our environment. And so the temporal association between the events, vaccination versus hygiene hypothesis, are very hard to deconvolute in my mind. It would be very interesting to try to directly test those things in an animal model, and to my knowledge, that has not been done in any detail.
Can I add one more thing? It's a very important question, and this is to ask Skip as well, is that in the context of immunizing against rotavirus, for example, is there any reason to believe that there's an unintended consequence of shifting the balance towards susceptibility, or not susceptibility, but it's shifting the balance towards prevalence of other viruses, such as norovirus, which are potentially, as you showed, associated with susceptibility to disease in the right genetic context. So is that an example where vaccination might?
I think that the answer is that the total burden of gastroenteritis caused by viruses has dropped significantly with the rotavirus vaccine, rather than there being the same number of cases now caused by a new agent. So I don't think that there is a shift in the medical or the population health importance of different viruses because you've eliminated one, but I don't think that it has transitioned over to there being more disease than caused by a second agent. Maybe other people in the room are more expert. Mark, do you?
No, that is correct. And I guess with respect to this question, it is worth knowing whether you think there's a critical time window with which any sort of perturbation would have an impact, because apart from hepatitis B vaccine, we don't provide vaccines to children before 2 months of age, the hepatitis B vaccine being administered at birth. Is it truly the first weeks that you're talking about?
I believe the mycobacterial vaccine around the world is given very early also.
Oh, it is, but certainly not in this country.
Mary?
I had a question about the persistent norovirus infection that you discussed. Are you suggesting that there are two separate populations of noroviruses, one that can produce persistent infection and others acute, or are there subpopulations within the norovirus that can do this? And do you have any information or evidence about where the persistence, in what level or what anatomic site you see the persistence?
So I can tell you what's known about the human situation. I can tell you what's known about the murine viruses. So in the human situation, beautiful work by a number of different people, spearheaded really by Mary Estes, who's now at Baylor, defined the virus and showed that it is an acute infection and that it is cleared in a reasonable time period. The symptoms are between 24 and really 72 hours, and it's cleared in most people, to the assays that were available in the last 20 years, within 7 to 10 days in most people. But there was this odd phenomena that it has no animal reservoir, like yellow fever would have an animal reservoir, but it was an epidemic virus. So where is it in between the epidemics exactly? And so more recently, much more sensitive assays have defined that there is a subset of apparently immunologically normal individuals who shed noroviruses for long periods of time. The question of whether that is a viral contribution because they're different strains, or is that a host susceptibility issue, is not defined. The best evidence for susceptibility to human norovirus disease is actually blood group antigens. Certain people have different blood groups, they're secretors or not secretors, and if you don't have the receptor, you can't be infected with some of the strains of norovirus. In the mouse system, we have persistent and non-persistent. We've mapped it. It's to a single amino acid. I didn't talk about it. In a protein of unknown function, we can specifically convert viruses to be either persistent or persistent with that mutation. We can also make non-persistent viruses persist by deleting the interferon receptors in either dendritic cells or macrophages. So there's a host interaction where something in the innate immune response is controlling persistence and something in the virus is controlling persistence. I would say that across all viruses where persistence has been studied, it has been both. LCMV, herpes virus, measles, all of the contributions to persistent virus infection are dual contributions that I'm aware of between something from the host and something from the virus controlling that knife edge where you're kind of on a balance over long periods of time.
Claire?
This may be a somewhat off-the-wall question, and the thought originally came to me as I was listening to Larry talk. So in part, Larry, this question is to you, but I think really I'd be curious to get thoughts of anybody on this afternoon's panel. And that is, you were describing the different community types in the human vagina and how they are very dynamic and they change, in some cases daily, based on factors we probably know something about, hormonal status, diet, and other factors we perhaps don't even fully recognize at this point in time. And the question occurred to me was whether or not you've looked or plan to look at coordinated changes not only in the vaginal microbiome, but also in other environments in the human body. Enterotypes for the human gut microbiome have been described, and I'm really curious, as we start to think about the microbiome and take a more holistic view towards health and disease, should we start to get away from the very natural tendency to look at these in a specific location associated with diseases where we think there should be some correlation, to really be looking at the microbiome in health and some of these disease states more globally?
Me first, huh? Well, I don't know. First of all, I'd like to say I think that the changes that we're seeing might be very misleading. I think that when we get to the functional analysis of the metabolome and the transcriptome, that it's going to be greatly simplified. And understanding a little bit more about the metabolic interactions is going to hopefully identify key interactions, critical nodes in our ecological network that might be important. I don't know. I can't in my mind make a link to the kinds of changes that might occur, say, in the oral microbiome and associate those with the vagina in the absence of sexual activity. But I could see doing studies where there was a concurrent analysis of both the gastrointestinal community and the vaginal community, because I think that one of the remarkable things about the vagina is they're probably continually being invaded by fecal organisms, and changes in the relative abundance of those or the appearance of certain organisms might have a dramatic effect on the vaginal communities. But you can add anything else?
I'll just add that, I really think we have to begin to think of disease itself as, or get away from speaking of one etiological agent and speak of microbial communities and the patterns and changes that go on to create environments that are more susceptible or more conducive to a particular organism taking hold and then causing. So I think that Larry's right. It's changed very dynamically, but we've got, I think, have to move towards speaking of diseases as patterns of microbial communities relative to the pathogenesis instead of just one organism.
So there's, I thought that the question has to do with whether we should look at everything everywhere versus whether we have localized responses. And I think the answer is both.
That was the question, and that was a bit of an oversimplification. But I guess really what I was getting at was that we do tend to focus on a disease, and we think that, okay, IBD is a great example. Let's just look at the gut. But maybe there are things happening elsewhere, and I guess I'm just wondering that we're sort of perpetuating some of this, what we've been doing for a very, very long time, one disease, one organ system, and I'm starting to see this happening now with the microbiome, and I'm just throwing out the question, should we be thinking a bit more broadly?
So, Claire, I think there are three things that are happening now that I would point out, which two argue one way and another argues a different way. So, for instance, you saw from Paul a whole set of things that argued that the immune response was different in different places. There's a really pretty set of very high-level, good papers showing now that you can get local immune memory. I mean local. Like, if you put a virus here, there is immunity there that is long-lived, but there's also systemic immunity. So that would argue that they're very local phenomena. It's clearly true that the immune molecules as well as the microbiome are quite site-specific, so that would argue we should pay attention to local environments. But then on the flip side, all the phenotypes that I showed are systemic phenotypes. And there's some very nice work which has been done, and you'll hear some more, I think, tomorrow, but by an investigator named Akiko Iwasaki at Yale, who's shown that gut microbiome, she argues it's the gut microbiome, alters the fundamental nature of CD8 T cells to influenza antigens in the lung. And so that really is a transitive property from one organ site to another. So it doesn't make any sense to study the lung if it's really that there are pattern recognition receptors in the gut, which is how she thinks it's working, which are then sending signals all the way to the lung to make you make or not make CD8 T cells to the virus in the lung. So probably the answer is you have to be integrated, but you can't ignore your neighborhood.
I'd like to add a little bit more to this, at least in the case of the, the vaginal microbiome. Our thinking has been to go the other way, and that's to look at a very much a localized environment. I mentioned in my talk that the vagina in many ways operates as a plug-flow reactor, and in engineering terms that means that the substrate comes in at one end and it's converted during a process, and then a product is obtained at the other end. That is, in fact, the way the secretions of the vagina work, and that the mucus is produced, secretions are produced near the cervix, and then they flow out. It also has rugate throughout the entire thing. What we sample then is just the mucus, the vaginal secretions on the surface with a swab, and I wonder if there's even more structure, the way it's been seen in the gut, to where it's in the crypts, or it's not in the crypts. And when you say it's an anaerobic environment, that's probably true for much of it, but not all of it, because of the effect of the blood circulation and the oxygen there. So I think we might want to zoom in a little bit and see who's talking to who at what point in the system. Because I think we're making the same mistake that people would make anywhere where you say that the vagina is somehow a homogeneous, monolithic, one environment. And I think there are many environments that might make a difference exactly who's where and what's happening at a local level, rather than going the other way and thinking about how does something that happens on your skin affect your vagina.
There was a question over here at the wall that we'll come over to.
I have a question for Rick. It's been known, diversion colitis, which is when you have surgery and the part of the gut is now walled off, usually it's the colon, and then you develop a rip-roaring inflammatory bowel disease until you hook the gut up again and then the inflammation disappears. Have you looked to see if the CXCR16 mechanism and NK cell mechanism is operative in the human model?
We haven't done that. I actually don't even know if anybody...
Sorry, Rick.
We haven't done that. I don't know if anybody's actually even made a mouse model to create a diversion colitis. Maybe somebody else knows, but it's a very interesting question.
It goes to what Larry also said, because it does talk about dynamic flow of...
But it does speak to the point that there's an ecology that is that when abnormal can create an inflammatory environment. So there's no human studies I'm aware of, but you might expect that there would be whatever dysbiosis is. We don't even really have a good answer what the core microbiome or microbiota is, but there's some abnormality presumably in that context, and nobody's looked at that to my knowledge, but you would predict that there would be an abnormality there.
Keita?
So one of the things I was wondering is, why is it that you can't recreate the biota, the microbiome in the place, and the time has passed? I mean, it seems to me maybe Karen, in your model, you could, by putting these different pieces in place, or Forrest with your place, and see why is this such a time-specific thing is what I'm wondering, and why can't you go and recreate these systems?
I think one hint to an answer there, we've been looking at the composition of the microbiota over developmental time, as others have as well, and we see very distinct patterns. So I think, there are developmental changes in the host that really might change that host environment and foster different types of communities. And so then now, if you start off with a history of no associations and introduce at a later time point, that might just change the whole equation of how that colonization is going to play out, which is something that we can explore in our fish system.
I think as much as anything, I think you're absolutely right. It's a great, fascinating, important, critical question. And I would predict that, if you go back and look, 10, 15 years ago, when people start looking at, let's say, T-cell differentiation, and said that once you're a T-regulatory cell, you can never go back.
Yeah.
And now we know that you can T-regulatory cells be forced into a Th17 cell, or you look at the stem cell literature. Now we know we can take a pure, fully differentiated cell and suddenly figure out a way to force it back to become all kinds of different things. So I just think it's more, I apologize for seeming to be very rigid about this concept, but it's just our own ignorance. There might be a way to reprogram the system. Presumably there will be a way to reprogram the system, and it's up to us to figure out what that is.
But you might also have the problem. So the T cells, you can force them to bounce around, right, in what they're doing, but they would still have the same TCR rearrangement. So in some ways, you're catalyzed, right, in a true evolutionary sense. So you get exposed in different orders, and you get that different effect, no matter what your background was like.
Yeah, but evolutionarily, you would think there would be some way to recalibrate that system.
Yeah, but only on the next generation, not in the current generation, because now you've changed. It's not germline. It's all of the rest of the tissues, right?
I have another point to add, and it actually relates to what Margaret's question was. In many ways, what we're looking at are things that have happened in the last several decades of time, changes in the patterns of diseases and so on, which is just a sliver of time in the evolutionary history of the species. So in the way that the watchmaker might suggest, all the experiments have been done over time, and the limited set that remains are the ones that work for survival of the species. And so, the idea, maybe we can intervene in somehow and manage this system in a very effective way to sort of recreate what should have happened naturally. I hope we can, because the barn is on fire. And I don't know what else to do with it. But we are talking about trying to tweak with something that has developed over hundreds of thousands of years, with all the unsuccessful experiments becoming extinct. Right? And so it's an appealing thing, but I think our time frame has to be much longer and realize that the question is a good one, but the answer is why can't you do it? It's because it didn't happen before and it doesn't work very well. I mean, that's not true. That it's a problem we have to face, is that this isn't, even when I think about the vaginal microbiome, and I mentioned this sort of tangentially in my talk, this is a system that has evolved over a very long time to ensure the survival of the species. I mean, it must be very, very good at what it does, and with everything. I mean, the immune system, the nutrition, the bacteria, the colonization events, the successional events, and so on. The one thing that I do wonder about, in that same regard, is that menopause was never meant to happen, right? And everyone should have been dead at that time. And so I don't know how the rules of the game change when we've artificially, in many ways, prolonged life beyond what it would have been 1,000 years ago. Come on, Mark, go for it.
Can I just comment on that? You know, so one of the things, Larry, is that it's a life history strategy, and there are lots of animals that have So I won't go into evolutionary things, but there are some animals that are reproductive until the minute, you know, they just die. And then there are animals, particularly animals that have multi-generational training, that and it's not just mammals. I mean, it's all the way back. And so life history strategies are quite varied. So I wouldn't agree with that.
Larry, well, go ahead.
Let me take. I thought that was I understand that argument a little bit, but I disagree with it in the sense that that's the argument that within naturally generated signals, there's a certain set of deterministic responses that the organism can make, and we're trapped by our evolution. So I don't buy it, because we can make cells do things that they can't do naturally by manipulating signaling pathways once we understand them in ways that they wouldn't be naturally manipulated. So I think the idea that you can reprogram things is perfectly legitimate. I think that the argument that you heard here, which is that we've already rearranged all our T and B cell receptors so we're stuck, that's a little tougher.
Well, I have to jump into that, right? Because I agree with what you said, and I actually want to be more optimistic than I sounded. But the story that came out today that says, well, we have strategies for reprogramming things. They're called antibiotics, and so there are unintended consequences to manipulation, right? And so, and maybe I'm just playing a devil's advocate, playing on one side of it. But I think the idea that we can somehow manage this entire system so that it's like, wow, I don't know. Life histories.
Like not only life history strategies, but animals have tendencies to be specialists and generalists, and there are all these various ecological principles that apply to given species. That you can think about, and there are some animals that are extremely plastic, and then there are some animals that are not at all plastic. So luckily, I think humans are among the more plastic group. I think one of the things that Kata was talking about in this neonatal thing, I just wanted to make one comment about I think of the microbiome, as David knows, as the 11th the 11th vertebrate organ system, and it doesn't undergo development until after birth. And we always think of embryogenesis as being the big developmental period, but I think it goes on, and the development of that system doesn't happen until what we call post-embryonic.
And of course, some of these other systems also go on after birth as well.
Yeah, sure.
I was going to add my little 2 cents to Larry's sort of perspective, which would be to say we certainly aren't smart enough to think that we could deliberately reprogram the entire landscape. But I think one thing you could say is that if we are to be able to do it, it won't happen with one type of intervention, one form of manipulation. That because of all the interacting parts, there's going to have to be some kind of coordinated strategy that involves a number of new self-reinforcing sort of forces. And I don't exactly know how you start to tease apart the deliberate strategy that does that. But you can see where, just in a very simple way, this study in the New England Journal from Tufts that said, actually from WashU as well, that ready-to-use therapeutic food has a much more durable, lasting effect on malnourished kids when you pre-treat them with an antibiotic, which would seem a little bit odd until you think, well, maybe what they're really doing is really reshaping a little bit of that landscape at the time that you deliver this one, factor, in order that it may have a better chance of even sort of a short-term, more significant effect. So that might be the sort of the principle involved, but exactly how you do it. There's some questions over here. Angela.
Yes, I wanted to make a comment in response to Larry, saying that postmenopausal women only existed in post-20th century. Actually, there's a beautiful study going back to the 1300s on the British aristocracy. And this demonstrated very nicely that elderly grandmothers were enormously important to the fitness of their grandchildren. Postmenopausal women have been with us, I believe, for a very long time, at least in the British aristocracy. and although certainly many people died earlier, there have always been elderly people, and particularly elderly women have been very important to the survival of their descendants. I would hesitate to call that being for good for the species. So that's just a comment of fact. I would like to make another general comment. Sorry, did you want to respond to that, Laurie?
No offense intended.
No, that's all right. I'm sure there have been studies of people other than the British aristocracy, but that was the one that came to my mind. The second point I wanted to make was that in this discussion, we've been focusing entirely on how this or that microbiota, be it viral or whatever, is good for the host. We have to remember there's another player in here, and this is the selection pressure on the microorganisms, in many cases whose existence depends on utilizing their host. And an animal host is a nutrient-rich patch and a means of dispersal, and some of the traits that we see may not have evolved for the benefit of the host, but for the benefit of the microorganisms. And that's just a general thing, and we must be careful not to be too Panglossian about all of this, that our interactions in a so-called healthy animal, I hesitate to use that word now, but in a healthy animal are necessarily for our good. It could be for the good of the microorganisms, and that's just me getting on my soapbox a little bit.
Yeah, I'd just like to respond to that, and I think we're seeing some hints of that as well in our analysis. So if you'll recall, we saw these different trends of different bacteria having different fitnesses in different contexts, and it seems that, for example, this Vibrio really was able to thrive in a more neutrophil-rich environment or inflamed environment. So we think that, and that was in contrast to some of the other organisms, and that's the interesting interplay between the members of the community, but also how their impact on the host then impacts their fitness. I think is something that is clearly emerging.
And that actually, just want to say one more thing, which is it could well be that in some instances, what we're seeing is the animal host or human host is actually addicted to their microbiota. We don't get any benefit from some attributes, but they've been with us always, and through natural selection or what have you, that we can't do without them, not because they're beneficial, but because they're always with us, and our signaling systems, metabolic systems have just evolved to be associated with them, and we should actually discriminate quite carefully between these instances of addiction versus instances of benefit. And I think your zebrafish system may well be very helpful in elucidating that.
Thank you, Angela. Over here.
Hi, this is Simeon Zhou from the National Heartland and Blood Institute of NIH. Just prompted by your discussion about early life vaccination and also hygiene, that's the medical development you may have noticed or not. That's the transfusion into recipients younger than 1 year old. Now in the U.S., every year we have over 50,000 such transfusions, including the perinatal transfusion, including the transfusion to a fetus. So I'm sure, this blood, I know this blood has been screened for many infections, more stringent screening compared to transfusion to other regular recipients. But still, I'm sure there's a lot of stuff in the blood that gets infused into the fetus, into babies. So I'm sure there's there's going to be some kind of impact, immunity priming, immunity maturation, and other things. So I'd just like to have your expert opinion on that.
So if you take a viremic blood, just from volunteers that are, as far as we can tell, are healthy, you only do DNA viruses, not even the RNA. You come up with anywhere between 20 to 50 new viruses every time you do it. These would be people that would be screened as not having any viruses in them. They're screened out.
Just so I can say one thing that's not virus-centric. The other thing to note is that while the MHC, the major histocompatibility locus, is what we mostly think about, the fact is there are many, many, many minor histocompatibility differences between people. They're just not screened against in blood transfusion. So the other thing that you would have to think about was that it may be that exposing the very young immune system to a large variation in allelic structure of proteins outside of the MHC could also have some effects on the repertoire that you could develop. So I think it isn't just about what might be there that we don't know, but there's the issue that you're exposing them to a lot of proteins that are not truly self, when you do that.
But also early in life, there's this concept of tolerance. In fact, if you go back to the original studies of the development of tolerance, that go back to the 40s, and you look at where you have sibling calves that are during early placental life that are residing in the swing placenta, that they're fraternal and they cross-tolerize to each other. So you can, and that process of cross-tolerization also continues on for a certain period of time after birth. So there may be some, not to say, not everything is necessarily detrimental. There might be some inadvertent beneficial aspects of it as well, but they're non-infectious.
All right.
Yeah, thank you. And also, just final comment, we do have a special PAR in transfusion medicine. So I would really much like to see more applications to our PAR to address this and other issues. Thank you.
Yeah, I very much enjoyed all the presentations. Being in the military frequently, I like to come back, if I can, from these meetings with ideas for application for all the really cool things we've thought about and discussed. As was already mentioned in a previous forum, we sort of focused on the issue of getting away from the war paradigm. And there's no question, a number of us in this room have dedicated our lives to making, vaccines, antivirals, antibiotics, et cetera. And yet it would appear that, we're sort of losing that battle, in a sense, as resistance grows, et cetera. And something I've asked some previous groups is what are some potential applications from some of the things we've discussed? Are there potential ways we can reset the microbiome for patients as opposed to go after a directed, pharmacologic intervention?
Here's a crazy idea. My second crazy idea. I've thought about the idea of having women bank their own vaginal secretions. And so the probiotics that are currently used, vaginal probiotics, are in fact dairy organisms, and they're not among the species that are found in the vagina. And as are the other organisms, they're uniquely adapted, selected, or whatever from a given individual or within a given individual. And the idea of having, it's sort of the fecal transplant thing, only with a new twist on it, and that's that you literally bank your own secretions, and when you have an episode of BV, that you re-inoculate yourself with those. They would be frozen banked, but that's one thing that I think might be the most interesting thing to evaluate, whether or not it would be an effective treatment for bacterial vaginosis.
Yeah, well, okay, Skip.
Go for it.
I was just going to say that tomorrow we have, at the end, in the afternoon, we have actually a couple of talks on this whole topic of where are there potential applications, and Michael Fischbach, others are here. So that's another, and some of us have talked about banking other part early in life.
So a comment on that. I think that the conceit within the question and amongst many of our talks is that we have actually defined the microbiome. And the fact of the matter is that we have some virologists here and some bacteriologists, but there's a huge underrepresentation of the fungologists. There's beautiful data suggesting that fungi may, in fact, contribute to certain inflammatory processes. Most humans in the rest of the world have apicomplexan parasites, which live in them. There are worms. So I would just put out that there's a sociologic issue. Bacteria frighten me. They're so complicated, and viruses are so simple. I think my bacteriologic colleagues are deathly afraid of actually thinking about the complexity of the viruses, so they sort of look this. And to be honest, part of the application of the new technologies that are rising, in my view, should be that we actually define the whole thing. What we've learned is that when you look at the bacteria in huge detail, you learn amazing things. But right now, I think we're short of application because it's the elephant in the room, and we're just touching the left hind leg of the elephant. And you see that the microbiology expertise is very isolated in silos, and that there is a major issue here. If we're going to think about the metagenome, then we can't actually think about the metagenome with a virologist telling people. I mean, there has to be an interaction across. So I suspect that there's a major application in defining human health that has to do with having a much more comprehensive RNA plus DNA assessment of what we actually are as a genetic organism.
Yeah, just I really appreciate what you say, and there's no question. I mean, we're continually confronted with our ignorance and the vastness of that. Within the military and forest, this will warm your heart. We're looking at the use of phages in wound infection treatment and putting a real effort in there. And I can imagine after hearing your lecture that maybe we would look at some type of mucin treatment. I don't know. I mean, Larry, give me some support here.
It worked.
Yeah. Okay.
Yeah, I guess I just wanted to add that I think that we should really be listening carefully to how microbes are interacting with each other in these communities. And so, for example, I presented some data that, one microbe can have an anti-inflammatory effect when it's in conjunction with another. And we're starting to learn that some of this is mediated through secreted proteins. And so, I think just listening to what they have to say to each other could reveal all sorts of new exciting molecules.
May I?
So we also know, in ecology, where there's more of my life thinking about that, we know how to tear systems apart really well, actually, and so how you collapse one. We don't know very well how to put them back together. And in the cases we do, we know that you have to go through multiple succession stages. You should talk to Brendan and people like that, because eventually that will be what we have to do, I think. You're not just going to make one jump. If you really want to reestablish a complex ecosystem, it may be very complicated.
Michael?
I just wanted to comment a little bit, actually, to answer your question. It is, we know, coming from the antimicrobial peptide field, we do know increasingly greater amounts of information that help us, may help us control induction. So what you haven't really talked about is how the epithelium responds. Many epithelia respond following either colonization, infection, or injury with respect to their batteries of antimicrobial peptides, and I'll talk about that tomorrow. But we know, for example, that if you take certain rather simple chemicals like the short chain fatty acids or vitamin D, we can induce one or another antimicrobial peptides that can achieve a therapeutic effect. And I give you an example, the case of Shigella. So if you examine the colon of an individual, a human or a rabbit, who has been given a Shigella, good solid Shigella in dysentery, You see depression of the major antimicrobial peptides that are responsible for protection of the epithelial barrier of the colon. And with the resolution of the disease, you see restoration. It's been pretty well demonstrated that it's some sort of DNA, effect of the DNA of that organism that's somehow been inhibitory. But if you treat the animal, and trials have begun in Bangladesh, for example, with butyrate, the rabbit or the human has been, I think, with suppository or phenylbutyrate, actually orally, you see induction of colonic antimicrobial peptides, a number of them, and you see clearance of the Shigella, coordinate with, in kinetic relationship with the administration of this butyrate. I'll show you tomorrow that some sunlight can pretty much cure TB, and it's through a induction of an antimicrobial peptide. So I think you can play, I think the innate immune system, at least the epithelial system, and some aspect and some portions of the systemic system can be played with. But I had another question that's been bothering me, and the gut people up there from all different species could answer this one. It's obvious that we're being hit with obviously bacteria. Now, of course, the story of the viruses becomes terribly frightening. But what really has been disturbing me for some time is the fact that the nervous system of the gut doesn't have a blood-brain barrier. And the enteric nervous system, were it to go, would kill us, right? I mean, you can't have a non-functional enteric nervous system. You basically will not have a functional small intestine, and you will go for any number of reasons. And what's particularly annoying is that the fibers of the vagus track up into the dorsal motor nucleus and then go north. So there's every reason to expect that when a kid gets a viral infection, some of those viruses might very well be trophic for those nerves, and maybe this repeatedly happens to us, year after year after year, and eventually, some of our circuits go, which leads to the hypotheses of a number of people that Alzheimer's, Parkinson's, and perhaps other conditions are fundamentally of infectious origin, may derive from the gut, or the other portal of entry, which is the olfactory lobes. So are zebrafish a good model? Are there studies that could support, contradict, help us better understand the virome, the gut microbiome, and the threats to our nervous system or whatever?
Well, we do know that when we look at the enteric nervous system of our germ-free zebrafish versus our conventional zebrafish, that we see profound influences of the commensal bacteria on the development of that system. So, I think it's reasonable to think that there are impacts of microbes on that system. We haven't looked specifically at an infection model. I think that would be a possibility.
So it's been demonstrated in mice that certain viruses track directly up the vagus into the motor nuclei of the vagus or the facial nerve as well. Rabies being one example, but enteric reoviruses do this. So there is no barrier between your gut and things going up. So that's well documented. I think that there's no documentation that has to do with chronic neurologic diseases such as Alzheimer's. There is some data for maybe other viruses, but it's pretty controversial, I would say. There is, however, in Crohn's disease, we all talk about the inflammation and such. One of the diagnostic criteria for Crohn's disease is hypertrophy of the enteric nervous system. And so there are substantial effects of these chronic inflammatory processes on the structure and function of the enteric nervous system, which have not been studied in detail. It would be amazing if the same microbiome that people have studied don't have influences on the enteric nervous system. That would be a stunner. But if you ask for pathologists to evaluate transmurally what happens, The answer is that a very substantial signature of Crohn's disease, for example, is hypertrophy of the neurons of the enteric nervous system.
And there is a lesion named after you. That is
So the virgin lesion?
No, the skip lesion. that remains still, right, difficult to understand.
So there's actually even I hadn't thought of it before, but there is an enteric nervous system knockout animal that develops spontaneous inflammation. The mechanism is not known. So it's interesting that they're virotropic nerves causing some of these disorders.
We have just actually 5 minutes left before we're going to need to wrap up. Todd, you have a question?
I'd like to go ahead and shift the focus from the gut to the lung. How important are phage in controlling the bacterial population in the lung, especially in cases of cystic fibrosis and other chronic infections?
So I'd like to tell you they're really important, but as far as we can tell, most of the time when we're looking, and so in a healthy lung, we're doing a pretty good job of keeping it clean. So what we're seeing more is just the stuff we're breathing in being pushed back out. In cystic fibrosis, we see something even crazier, which is that during the disease flare, there's a lot of viral activity going on, and they're moving a lot of different antibiotic resistance genes around, things of that nature. But when they're in that what we call the climax community, when they're just in those herbal biofilms, there's almost no phage, and and we get the feeling that the biofilm is keeping the phage from moving from one microbe to the other. It looks something of that nature. The only viruses that we see for the most part tend to be the like herpes viruses and so forth. So it's almost like things coming out of the human system from the tissue damage. The phage seem to be all temperate, and they seem to be behaving as temperate phage at that point, not politically.
Is it just because there aren't enough of them?
No, there's a lot of them there. They're just not killing well. So I think biofilms are doing exactly what biofilms are supposed to do in some cases, which is keep the phage from moving around and killing the microbes.
Oh, Forrest, that might be the only time I've ever heard you say that phage weren't doing much. That's saying something. I think this might be a nice time to wrap it up for the day. It's been a lot of great talks, even better discussion. I'm hoping we have equal or more amounts of time tomorrow to talk about, again, some recurrent interesting themes. Thank you all for your contributions. Thank you all to the audience and to the rest of the forum and to staff. Let me just point out that the schedule had said that we were going to continue till 6, and then